In this, hello guys, and finally, today we are going to be doing a science and biology video. You math people were like, okay, I'm finally posting math videos, but now we have to give biology some love. And I'm gonna give genetics some love because it's my favorite biology. And so we're going to be talking about gel electrophoresis. Now, gel electrophoresis is probably one of the most useful processes in all of, not even in all of genetics, but in all of biology. I mean, it's how we isolate DNA samples. It's how we it's how we use PCR. It's how we can do DNA replication. I mean, it's almost used in every research analysis biology thing. It's used in that. Like, you'll see what I'm talking about. It's such a useful process. And so, what is gel electrophoresis? Well, in a nutshell, gel electrophoresis is basically the product of breaking down DNA fragments. And so specifically, we will be talking about how we use gel electrophoresis to create a restriction map. Now, a restriction map is essentially how we separate our DNA fragments via gel electrophoresis. So basically, let's say I have this chromosome, right? And I have these restriction sites, like those little lines are my restriction sites. Now it looks like a finger, but let's just say those are my restriction sites. Well, things called restriction enzymes can bind to the restriction sites and essentially cut up the DNA, or essentially the gene of interest. It can cut it up into fragments. Now, obviously, these fragments, there's thousands and there's hundreds and thousands of fragments, but for the simplicity of this video, I'm not going to draw 1,000 fragments. But what I'm saying is that essentially, to use gel electrophoresis to create a restriction map, we first need restriction enzymes, which are enzymes that cut up things. Like in genetics, you just refer to it as enzymes that like literally cut up or chop up stuff. So in this case, we use restriction enzymes to bind to their restriction sites. So let's say this is a chromosome, a haploid chromosome. I know this chromosome is not really like a chromosome, but I'm not good at drawing. But let's just say we have restriction enzymes. So maybe there's like a restriction enzyme here, another restriction enzyme here. And let's say that they bind to this chromosome and essentially break it up into DNA fragments. Now, how do we actually separate these fragments due to their respective size? Because that's really what a restriction map is. A restriction map is essentially just a map used to essentially separate these fragments due to their respective size. And why is this useful? Well, here, if we're trying to isolate a particular DNA sequence, for example, in southern blotting and northern blotting, we would try to isolate like a DNA or RNA sample, respectively, and we want to examine what gene sequences there are in that DNA or RNA sample. But so it's very useful for us to actually separate these regarding to their links, because it makes it easiest. It literally makes it easier to determine what are the gene sequences of these fragments. If we can just separate them by length, we can easily identify which one, because we obviously know that if the gene of interest is the longest one, then if we can find some way to separate this by length, then we can easily just identify the gene of interest or the fragment of the chromosome or DNA that we want to examine. So we want to separate the chromosomes by length so we can easily be able to identify gene sequences. You'll also see gel electrophoresis in connection to southern blotting and northern blotting, and I might be doing a video on that. But in this case, that's why restriction maps are extremely useful. We don't just do stuff because we want to do them for fun. Everything actually has a reason, so it's good that you know the reason. Just like, analogous to knowing a proof in math, for formula, So essentially what happens is that these fragments get broken up. And now, how do we actually separate these fragments? Well, to do that, the first thing we need is a gel. Now, there's two types of gel. There's agarose and there's polyacrylamide. Now, I might be pronouncing it wrong, but essentially the difference, let's just say this is the gel. I don't really care about how it's supposed to look. You can probably get better diagrams on yourself from Wikipedia, but there's two types of gels, right? Commonly used in gel electrophoresis. You have agarose and you have polyacrylamide. Now, agarose is simply used when the fragments are like really big or really large. Like if the gene of interest or the fragment that we want to study is really large, then we're probably going to use agarose. If it's small enough, like we can probably use polyacrylamide. So if the fragment is essentially like small enough for polyacrylamide, or it's not too big for polyacrylamide, we're probably going to use that. If it's too big for polyacrylamide, then we'll just use agarose gel. But in this case, we won't focus on which one we're going to use, because you'll actually see in other processes such as Western blotting, we actually use polyacrylamide to put the protein samples instead of agarose, because the proteins aren't as big. As if we needed an agarose gel. But don't really worry. Here we're kind of just focusing on slab gel electrophoresis to create a restriction map. It's kind of more of an overview in this. So basically we need this gel, right? And we're not going to specify which gel we're going to use, okay? We're going to need this gel. And let's say we label this A, B, C, and D. Let's say I want to find B, right? What we're going to do is we're going to take these fragments we have over here, and we're just going to transfer it into the gel. Now, think about it. How are we going to, after we do this, like, what are we going to do now? Like, we've transferred it, and we've done all of this, and I really don't know what to do. Like, what should we do? Well, look, what we can do is we can create this. So let's just say we erase this. Let's create the new product. We have this this gel over here, and we have our fragments, right? Let's say this was a B, in no order. I don't really care, but we're trying to find B, right? So let's say this is the product. Now what we need to do is we need to create an electric field. The reason we want to create an electric field or a field of charge is because first, we want to create a negative and positive end, and you'll see why. There's actually a very good reason of why we want to do this, and I'll explain that in a bit. But the first thing we need to do is to create an electric field. To do this, we can connect this product. So after we transfer the fragments to the gel, we can connect this to a voltage source. So maybe there's a battery over here. So that, let's say this is the battery. And what this battery does is that it creates a voltage source. And so essentially, it creates two sides. It creates a cathode side and an anode side. Now, the cathode side is going to always be negative, and the anode side will always be positive. Now, let me tell you why this is useful. Well, assuming we're working with DNA, right? We know that DNA is very negative. Why? Because it has many phosphate groups. I mean, it literally has a sugar phosphate backbone, an alternating sugar phosphate backbone. So it's going to have tons and tons of phosphate groups, giving its negative charge. So since DNA is negative, we know that if we connect this electric field to, let's say this is the apparatus or the product we already made beforehand, if we connect this to this a kind of electrophoresis setup, then what we can do is create two sides, a positive and negative side. And we know that since DNA is negative, then it must attract to the positive side, which is the anode side, which means that the fragments are going to connect to the anode side. But since we know this, why is this so useful? But let's just think about it. We know that the gel is porous, which means that, I should have probably specified that before, but the gel is porous, which means that it has pores in it. So we know that, just think about it logically, right? If a gel has pores in it, right? If a gel has pores in it, like that means like tiny holes for the fragments to pass through, then the larger fragments are probably going to pass through more difficultly in the pores than the smaller fragments, right? Because if something's like small, like if I just say, get through this hole right here, let's say I make a small hole, and this is a big fragment, and I make the same size hole here, and say it's this small fragment, well, I mean, it's obviously easier for the smaller fragment to go through this hole compared to this fragment over here. So we can use this kind of same analogy to say, well, the larger fragments are essentially going to move slower to the anode side than the smaller fragments. So the smaller fragments will move through the gel more quickly simply because they're smaller, because of their physical size. They can move through the pores much more easily. I mean, obviously, if you're trying to move through like a confined space or like a hole, I mean, if you're smaller, you're gonna have an easier job to move through that hole. So if we note that the smaller fragments will pass through the gel more quickly, and we also note that DNA is negative, which means that it attracts to the anode side, then all the fragments closest to the anode will be smaller than the fragments closer to the cathode. So since we know that B is the gene we're trying to isolate, or the gene we're essentially trying to look for, what we can do is we can essentially see that B is going to be closest to the anode side. To the cathode side, or furthest away to the anode side. So this will be B, and let's say A is the next biggest, and then C, and then A. And so basically, from here we can say that since this gene is closest to the cathode side, then it's the biggest. And from here, if we know the relative size due to this gel electrophoresis setup, we can easily create a restriction map, and then we can basically say, okay, this gene is X base pairs long, and just create a map. And so that's basically gel electrophoresis. That is the kind of intuitive feel for gel electrophoresis. And there's many applications of gel electrophoresis. There's SDS polyacrylamide gel electrophoresis, which is used in Western blotting, and there's many other processes. But this is just an overview of using gel electrophoresis to create a restriction map. And I didn't have the best videos, but I kind of just wanted to show you how that is done.